Good morning. My name is Ricardo Sandoval, and today is Saturday, June 20th, 2020, and this is the results video for lab number 10, Antibiotic Sensitivity, Kirby Bauer Diffusion, for Biology 225, Microbiology for the summer 1 term at Los Angeles Pacific University. And here's just a quick sign that I'm just summarizing everything that I just said. So, to recap, what I did in this lab was I'm tested how resistant or not resistant the microbe Staphylococcus epidermidis is against 3 different antibiotics: penicillin, novobiosin, and gentamicin. So the experiment itself was achieved using a 9mm Petri dish through which I poured and cooled agar. Once the agar cooled appropriately, I then added 8 drops of my epidermidis culture using a sterilized pipette. I then spread that culture evenly around the agar using an L-shaped spreader, and then let the agar plate sit for about an hour or two to ensure that the culture itself had properly absorbed into the agar. At the conclusion of the waiting period, if you will, I then added a disc each of gentamicin, novobiosin, and penicillin to 3 distinct areas as marked on the exterior of the agar plate or petri dish with a permanent marker, a Sharpie. My agar plate then sat for a total of 24 hours. Since after 12, I saw no growth, no activity, and then here we are today. So the expected results of this experiment is that I get kind of what's called a lawn, basically kind of like a white, filmy substance, and then I will perhaps have what are called zones of diffusion, kind of like circles around, either indicating if there is a zone of diffusion, basically an area where there is no microbial growth, that the particular antibiotic, or rather epidermidis, is inhibited by that particular antibiotic. If there is no zone of diffusion, that means that Staphylococcus epidermidis is resistant to the antibiotic. So in medicine or in a clinical setting, if an antibiotic is resistant, if a microbe is resistant to an antibiotic, it can't be used because the antibiotic will do nothing to kill it. Versus if a microbe is inhibited by an antibiotic, we know that it will stop the growth or kill it, so that antibiotic can be used for treatment. So just to kind of rehash some vocabulary that has been used in this laboratory. So we have bactericidal and bacteriostatic antibiotics or agents. So something that is bactericidal kills a pathogenic cell or a microbial cell through a process called cell lysis, while something that is bacteriostatic basically stops the growth. So it stops DNA replication, and then as cells can't divide, the microbe itself will die. So then we have the difference between wide spectrum and narrow spectrum antibiotics. So something that is a wide spectrum antibiotic kills everything. Common examples are like clindamycin, rosefin, zosyn, for example. The 3 antibiotics used in this experiment are narrow spectrum, so geared to a specific type of microbe. Then we have gram-positive versus gram-negative bacteria or microbes. So the most significant difference between something that is gram-positive and something that is gram-negative is a membrane called peptidoglycan. So those microbes, like Staphylococcus epidermidis, that are gram-positive have a very thick wall of this peptidoglycan, so they will grab hold of, for example, a stain or they'll grab hold of an antibiotic. So the bactericidal or bacteriostatic property of the antibiotic can take place, versus something that is that is gram negative. That membrane, the peptidoglycan, is much thinner in comparison to something that is gram positive. So because we have that thinner wall of peptidoglycan, it does not grab hold of a substance or a stain as easily, and those microbes themselves can be a bit more difficult to kill. Okay, so that's some vocab I've summarized. So now I'm going to show you my results, and unfortunately, these results are not what I was expecting to see. So here is my agar plate petri dish 24 hours after incubation. So again, the PNC is penicillin, the NVB is novobicin, and the GNT is gentamicin. So, as you can see, or as can be seen, there is growth of the organism or the microbe Staphylococcus epidermis throughout the novobicin and then the penicillin, and then in very minor amounts in the gentamicin portion. But what I was looking to see or hoping to get was something like this. So that white area is kind of the lawn, and then those areas where you can pretty much see through the plates, and then you have the antibiotic disc. Those indicate microbial inhibition. So whatever these antibiotics are do inhibit the growth, and then further and further out, they don't. So as I stated in my experiment video, this is kind of like the same premise as a nuclear bomb. So the place where you drop the bomb, ground zero, you'll have the most destruction, or it'll be most barren, and the further and further and further you go out, structures will still be there, plants will still be there, animals, people, things of that sort. But again, I don't have that result. So the hypothesis that I had come up with at the initiation of this experiment was that Staphylococcus epidermidis would exhibit resistance to novobicin and would be inhibited by penicillin and gentamicin. However, again, given this, there's no way to test whether or not my hypothesis is invalid or valid, whether it's valid or it's to be nullified. So, throughout my experiment, and you can see in my experiment video, there was nothing that I did to contaminate substances or improperly place things. So, and obviously, we do have some microbial growth, and I did spread the microbe evenly around the agar. So I'm very much at a loss why I got this instead of this. So had I gotten this, what I would have done is I would have measured in millimeters, basically from this zone of diffusion, this clear part, to the antibiotic disc. And of course, the larger that distance, the more inhibition, or I guess the stronger, if you will, the antibiotic is, versus something that is smaller, the weaker the antibiotic is. If, for example, like in this case, right here, as you can see, there is no zone of diffusion. Whatever this antibiotic is has no inhibitive properties against this bacteria, so this bacteria itself is resistant to that antibiotic. So a relatively short and sweet results video, unfortunately. There is a second part that has to do, again, with this slide, the penicillium with conidia. That is a spore, and it's asexual, but there really isn't much for me to show you as far as the results video, because it involves taking pictures of this with a microscope and answering some questions related to how this penicillium rose works, economic value. So you'll see my answers or results to that on my final written report. So this is Ricardo Sandoval again. This was the results video for Lab 10, Antibiotic Sensitivity, Kirby Bauer Diffusion at LAPU for the summer 1 term for Biology 225 Microbiology. Thank you.